Hello, my name is David Sutterer, and I'm a postdoc at Vanderbilt University working with Jeff Woodman. And so first up, I just wanted to say thank you for taking the time to tune in for my VVSS talk. And so, as you all know, visual working memory is a severely limited online mental workspace. In order to optimize the limited capacity of this system, our brain needs to figure out a way to efficiently select and maintain relevant information, while also finding a way to ignore irrelevant information that's constantly present in our environment. And so one mechanism that allows us to do this is covert spatial attention. And so the project I'm going to talk about today is motivated by a vast body of work that links human EEG activity in the alpha band with covert spatial attention and working memory. And so this relationship is nicely illustrated by the patterns of alpha band activity we can record during the delay period of lateralized visual working memory tasks like the one that you see here. And the kind of specific canonical example is when we cue an observer to remember items on the left side of the display, we typically observe a decrease in alpha band power over posterior electrodes that are contralateral to the target items, while at the same time we observe an increase in alpha band activity over posterior electrodes that correspond to the location of the distractor items. And so, perhaps not surprisingly, this has led to the proposal of two distinct functional roles for alpha band activity and covert attention. And so one idea is that the decrease in contralateral alpha band activity relative to targets reflects target enhancement, while the opposing idea is that these ipsilateral increases in alpha band activity reflect inhibition of the distractors. And so this debate is ongoing, in part because it's really difficult to distinguish between these two types of accounts with balanced displays that don't provide very much information about how attention is allocated to individual items. So one way that we might advance this debate is by considering this growing body of work that has shown that in tasks where observers are asked to remember the location of items presented around fixation, that we can actually use the pattern of alpha band activity across the entire scalp to reconstruct remembered locations throughout the delay period. And this suggests that there actually might be more information in these patterns of alpha band activity available to us than we can typically take advantage of in these lateralized designs. And so in the experiment I'm presenting today, what we did is we combined both of these approaches in a single task design that'll allow us to simultaneously measure lateralized alpha band activity while we also track the location of individual targets and distractors in the display. And so the task is pretty similar to the lateralized design that I just showed you, except that we made some tweaks that allow us to fully counterbalance the location of both the target and distractor stimuli that we're using in these types of displays. And so here's an example trial. And so on each trial, we, like before, cued observers to remember items on either the left or right of fixation, and then we simply ask them to remember the location of one or two colored dots for a brief delay. And then at the end of the delay, we cued observers to report the location of one of the target dots that they saw by having the fixation turn either blue or green. And so the goal of this approach is to better understand how alpha band sensitivity to targets and distractors differs. And so specifically, we're focused on kind of two categories of questions. So the first thing we want to look at is that based on previous work, reconstructing remembered locations was always conducted on stimuli presented across hemifields. And so the first question that this approach allows us to test is whether alpha band activity actually tracks individual target locations within a hemifield, or if instead this global alpha signal is limited to the angular location of stimuli around fixation. And then the second question that this approach will allow us to test is whether or not alpha band activity tracks the suppression of individual distractors, or if, to the extent that alpha band activity does reflect suppression of distractors, if this kind of suppressive mechanism is more limited to broad swaths of the visual field, like a hemifield, or perhaps all unattended locations. And so getting into the results, our first question was whether we observed the kind of classic lateralized alpha effects with this new spatially balanced design. And to test this, we simply compared the alpha band power at the posterior electrodes contralateral to target items for one-item trials with the pattern of alpha band activity at ipsilateral posterior electrodes. We did the same thing for two-item trials as well. And then to get a nice, easily visualizable metric of these lateralized effects, we simply subtract the ipsilateral activity from the contralateral activity. And what we find is that we observe these classic lateralized effects with this new design emerging roughly around 500 milliseconds or so after the sample onset. This design, our next question was whether or not this global pattern of alpha band activity at the same time provides information about individual targets and distractors. And the way that we tested that question is by using an inverted encoding model. And so to just briefly describe the model, the way that it works is that we assume that the alpha activity measured at each electrode reflects the weighted sum of a set of location channels that are each tuned for a different location, we see here. And so just like any other sort of linear classifier analysis, what we did is we partitioned our data into a training and test set. And then we first use a set of trained data to calculate a set of weights that map between our predicted channel responses for a given location and the observed pattern at each location. And then once we know the relative contribution of each channel to the response of each electrode, what we can do is we can take a new set of data and work backwards using the pattern of EEG activity on the scalp to estimate the channel responses when each location is remembered. And so if our model works, we should see something like the response here, where when an observer is remembering a stimulus at 120 degrees, we should see a peak channel in the channel corresponding to 120 degrees, as well as elevated responses in the adjacent channels. And so our first question can actually just be answered by looking at the average channel tuning functions for each remembered position after we average across the delay period. And so in the plot on the right here, what I'm going to be showing you is a bird's eye view of the channel tuning functions that I just showed you, average across the delay period. And so we have stimulus location here on the y-axis, and then the location channel here along the x-axis. And so if alpha band activity tracks the individual target locations, what we'd expect to see is a peak channel response in the location channel corresponding to each stimulus location, so kind of a nice diagonal across this plot. And alternatively, if alpha band activity instead tracks the angular location of items relative to fixation, what we'd instead expect to see is pretty clear, nice channel tuning functions at 300 degrees and 60 degrees, where there's only one angular location in our display, and then relatively lousy reconstructions along the midline, where we've got multiple channels corresponding to kind of the same angular location. And so in particular, we'd expect to really not be able to differentiate between stimuli presented by fixation and further out the periphery. So what do we see? What we find is that we obtain nice, robust channel tuning functions for each position with a nice peak centered over the remembered location, and relatively little confusability between items along the midline, which suggests that these patterns of alpha band activity are selective for individual target locations within hemifield and not just angular location. And so just to double check that this structure is present in our data and not imposed by our model, I just wanted to show that we find the same pattern of results if we run the analysis a different way using a linear classifier and plot the confusion matrix or classifier outputs instead, suggesting that this isn't driven by the IAM, this really exists in our pattern of data. So our next question is how these alpha band representations for targets change as memory load increases and across time. And so here, rather than plotting the individual channel tuning functions, we can instead compare the spatial selectivity for the target item regardless of position across time. And so the y-axis here, instead of being a CTF, just reflects how much information we have about the remembered location. So higher on the axis means more information, towards 0 means you're at around chance. And so now the question is, how do these representations change across trials and set sizes? And what we find is that in line with our previous work, alpha band activity starts to track target locations relatively early, within a few hundred milliseconds of the sample onset, and then extends throughout the delay period of the task. Now, one surprising observation here is that location selectivity doesn't reliably decrease as set size increases, but one thing that differs here from past work is that we also have distractor items, so a possibility is that distractor processing might vary across set size and kind of explain these discrepancies. So in order to test this idea, we reran our model, and this time we trained the model on distractor locations and used that to determine whether or not alpha band activity tracks the location of individual distractors. And so when we look at our data, what we find is that At least for one-item trials, alpha band activity does track the location of at least an individual distractor item, while it's not above chance for multiple items. And so now the critical question here is, are there two patterns of alpha band activity happening at once? And so is there a pattern of activity for selecting locations or attending locations, and another pattern for suppressing those distractor locations? Or is this alpha band activity instead reflecting that observers sometimes accidentally attend to the distractors in the one-item trials when there's only a couple things on the screen? So the cool thing about this task setup is that we can test it by just simply running our model a different way. To do this, what we did is we ran our model again, and this time we trained the inverted encoding model on target locations and tested the model on distractor locations. And so if there's separate patterns of alpha activity for attending target items and inhibiting distractor items, we would expect that our reconstructions would be at 0 for those one-item and two-item trials. But instead, if these patterns of activity across the scalp reflect the same process of just attending the item, whether it's a target or a distractor, we would expect to see a similar pattern of activity to what we saw when we trained the model on just distractors. And so when we look at our data, what we find is that we get reliable above chance decoding for one-item trials. In fact, slightly more reliable and more robust decoding than we do when we train on just distractor data, which seems to suggest that this activity reflects observers attending to the distractor items rather than suppressing them. And so this provides kind of a tidy explanation for why we may not see a drop in alpha selectivity as memory load increases, and more importantly suggests that alpha band activity in these one-item trials isn't suppressing individual locations, it's tracking attention to the distractors. So to conclude, over the course of this project, we found clear evidence that alpha band activity tracks the precise location of target items. And while we found that we could track the location of a single distractor item, further analysis revealed that this was driven by attention to rather than suppression of the distractor location. So this suggests that if alpha band activity does play a role in suppressing distractor information, it operates on broad swaths of the visual field and not individual distractor locations. And to wrap up, I'd just like to take the opportunity to thank my advisor, Jeff Woodman, our funders, the rest of the incredible team in the Woodman Lab, and of course you for listening. And so please feel free to contact me with questions or feedback throughout the week, either via email or through my VSS presenter page. Thanks again.